We start with the introductions because now we have to have an introduction of the first speaker who will introduce the second speaker. So as you know, we're trying this new format. When the speakers give their presentation, we ask that a graduate student in the STC present for 5 minutes beforehand on a topic that is relevant, that'll be just come up in the speaker's presentation. So we're lucky, both talks today. We were also lucky last time. By Andrew, I think he's still reading his voice. No, he's over there. He did a really great job. And focused in on one or two of the topics. And this time it's going to be Ashwin Bale. He's from Rohit's group, as well as Seth, Kenkel, our main speaker today. And he's an STC postdoc, but he's also in the research group of Rohit Bhargava. And as you know, that's one of our main thrusts here, is try to develop an instrumentation that would allow us to measure a chemical composition throughout eukaryotic cells. And there are so many different kinds of chemicals there. This is going to be very difficult to do, but it's also a great use of machine learning. So he'll tell you about their combined sort of AFM, IR. I don't know if you'll touch on the machine learning aspect of it at all, Seth? No? That would be a third speaker that we would probably need for that. But to help resolve the spectra, because those who have a chemistry background, you know, a lot of the same molecules, well, guess what? They have CA, CC bonds and CN bonds. So there's a lot of similarity and then trying to be able to differentiate them. So now that I think Rohit is here, and we'll go ahead and start with Ashwin. You can please take over. Yeah, thank you.
Hello, everyone. So for today's talk on innovations in near-field infrared spectroscopy, we will mostly be covering, as I mentioned, about AFM-IR, which is mostly, obviously, atomic force microscopy coupled with infrared spectroscopy, which offers nanometer scale imaging approach, which can be combined with, obviously, the chemical information. To quickly show how this stuff works, you basically have your sample. When you shine IR light on it, there is a cantilever which hovers over it. Depending on the different vibrations in the bonds, you have different levels of absorption of the IR spectra and corresponding expansion seen in the sample. The cantilever basically measures that expansion, and then you can couple the topological information with chemical information. Obviously, why the instrument hasn't really taken off with respect to biology until now is because there's like many different problems involved. And to solve many of these, especially by controlling the magnitude of cantilever deflection, Seth has worked a lot during his time as a PhD student. I usually try to explain this to the best of my understanding, but since he'll be giving a talk later on, I'll sort of skip that part, which is basically the null deflection infrared setup. But what this really does is it gives you very high signal to noise ratio in terms of the images you see, and that really gives you the resolved spectra and deep chemical information in the cells. Overall, the talks will cover two parts, NDIR hardware development, which is the new AFM-IR setup which Seth is building, and the application side of which I am kind of involved with. So I will just give you one example of the application which we have been working on with the older setup until now. And in the context of QCB, obviously, it has been with regards to being able to resolve metabolites in single cells. Irrespective of the application, right now, the main kind of the hurdle in the initial part is sample preparation, which has to go through all these steps. Starts off with cell culture in hydrogels. You fix them, go through a graded ethanol dehydration process. You use epoxy infiltration process for epoxy to be able to infiltrate between the cells. Put it overnight, and then you have these blocks of epoxy with samples at the top here. Once you have these epoxy pucks, you go to an ultramicrotome and section them to about 100 nanometer thickness, which is sort of the thickness we work with for these AFM-IR setups. And you put them on substrates of interest. For example, this is just a silicon wafer shown here. We now work with gold wafers. So basically your 100 nanometer sections mounted on gold wafers, and you take them for imaging. Just showing one brief example of this experiment we worked with a few months ago regarding glucose localization. Basically, again, cells cultured in hydrogel. You use glucose as extra addition to the culture media. Uh, Obviously the normal glucose, and then you in this case we also added deoxyglucose, which is slightly different in the sense that you don't have an OH molecule here. And how this makes this different is glucose usually is broken down all the way to lactate, so it kind of becomes a bit difficult to track glucose in some cases, mainly because it might not be there till the end when we process the samples. How deoxyglucose is different is that it's broken down to deoxyglucose 6 phosphate, which sort of remains in the cytoplasm of the cells, so it cannot be broken down further. So with AFMIR, it becomes a bit more easier to track. So once we know the mechanism, you can acquire their individual spectra. For example, the orange one is D-glucose, blue one is deoxyglucose and deoxyglucose 6-phosphate. All of them have peaks in this sort of 1000 to 1100 range. And that's kind of good because epoxy has main peaks upwards of 1100, and then there's like main peak in the 1700 region. It also has spectra sort of in the 1000 to 1100 range, but it sort of starts to fall off, which is why you can use these two peaks simultaneously in reference to each other to make these comparisons. Once you have your cells processed using the standard protocol, you can take images. These are just images of U87 glioblastoma cells. 1660 is just protein distribution throughout the cells, which you see here. And then you go to the 1020 band, which should be glucose. So if you see all the stuff lighting up is primarily glucose, localized within different pockets in the cell. And just to make sure that there's no epoxy infiltration in between there, you also take a 1732 image, which should be the epoxy band. And you see most of these regions going dark except for a few pixels here. There's still work to be done to make sure this 100% is glucose, because you obviously might have some other contributions from other factors in the cell. But based on the experimental workflow, 1020 band should primarily give you glucose spectra.
Excuse me? Can you
Yes.
explain again what are you seeing in the 1660? What chemistry is revealed in 1660?
Yes, so stuff like 1660 is amide 1, so that's primarily the proteins which you see inside.
This is after the formaldehyde fixation, right?
Yes.
So can you give me a sense what does the formaldehyde do?
So it kind of denatures the proteins, if I'm not wrong, but it preserves them in their intact state, so they do not like die off and wither completely.
And what about the metabolites or whatever the other
Yes.
thing?
So that's something to be figured out, because when I use that protocol, it's for sure that not all metabolites are going to be preserved. For example, ethanol I use will dissolve most of the lipids, so for sure we cannot see any of the lipids in this case. But glucose does not go away, which is why in this protocol right now glucose can be figured till the last step. But if you're looking at like 10 other metabolites, first thing that needs to be make sure is through that protocol, do these 10 metabolites remain till the end? If not, then there needs to be some change to the original protocol to make sure they don't dissolve away. But right now, as the protocol stands, it's good enough for looking at proteins, glucose, and possibly other key bands like nucleic acids and stuff, which remain preserved. But yeah. And obviously, you can just put all these images together with respect to epoxy, and you can get this image where the red color should show you glucose localization. We can slightly go one step further to kind of get serial sections. So instead of just looking at one section, take 8, 10, 15 sections continuously and get images. So this is the first section, second, third, fourth, and so on. And similarly, this is again the protein band for 1660. And one interesting thing here would be, again, if you try to look at glucose, I'm not sure if this thing is consistent across all cells, but for this cell specifically, you can see the sort of the intensity disappearing away. So for this specific cell, through the depth of the cell, there is a change in glucose gradient as well, which you can see from comparison to the 1732 band, which is, again, absolutely dark. Having said that, obviously there are many challenges as we go through the whole protocol. Just highlighting one of them before Seth takes over is You have your cell oriented in the epoxy. So when you section them at such small thickness, you don't really know where your cells are. Because when we image these things, it's like a small field of view based on like a 40 micron by 40 micron area, which is very small as compared to these sections we take. And we don't really know where the cells are. So until 6 months ago, we were using TM imaging. So basically take a cell section, make sure that you have cells in there using TM, and then take the adjacent section for imaging. But the problem here was with TM, you can only tell if you have good quality cells, but you cannot tell if where exactly the cell is located, which again was a problem. Tried using infrared spectroscopy generally for some time. And this was good in the sense that you can really localize that there's like a big cell spot here at the edge of this section. But this really works well only for spheroids. Until now, we haven't had good signal for single cells when you have them at 100 nanometer section. So again, that was kind of a problem depending on what your sample is. Right now, what's working the best is autofluorescence, because I didn't really know this until a few weeks ago that 100 nanometer sections have a very strong autofluorescence coming. And this whole thing is the edge of a section. This thing is epoxy, which is completely dark. The boundary you see is of the matrigel, and then all the spots lighting up other cells. So with this, you can really tell where exactly to go when you have when you're just starting an fMRI image. Having said that, this was just the application side of things, showing one example about glucose-specific study on the old instrument, which we currently have, but Seth is obviously working towards a much better version of the current instrument, and he'll be taking over now to give much more technical details on that stuff. Thank you.
Right, can we ask another question?
Yeah,
Maybe
sure.
we'll go to the next talk, but,
No, you can ask Ashwin.
can you talk, what is the spatial resolution of your images? And what is ultimately limiting the spatial resolution?
And then it's right.
It's the electric field enhancement around the tip. So the tip's radius is something like 40 nanometers or so. So it's something like that.
So the tip determines the spatial resolution.
Yeah, we're actively exploring that. We have a third major collaborator in this, Aniruddha, who is kind of analyzing the resolution of the system with test targets, test polymer ramps, and edge measurements.
I guess related to that is how well do you register different images at different
That's a good question. I'll actually go through that, but I have a custom image registration, non-rigid registration code that registers the height images, which are all in theory, the exact same measurement. So we register those, and then we determine the deformation fields from that to remap the IR components. And this is an image from the original system, RPNAS work in 2022. You can see the registration is fairly decent down to like the 40 nanometer pixels. And I'll show an image later in the talk of what these look like before the registration, because over 3 hours of imaging each band, they do thermally drift quite a bit, almost hundreds of nanometers. So I first do a rigid mapping to them, and then I have a custom deformation field that I apply. It works fairly well, but that is kind of one of the main purposes of the new design system, is to image these faster, image the IR bands faster, and do it in parallel essentially. So that's kind of what I'll go through, so that you actually don't need that registration.
Maybe just before Seth starts, let me tell you, he's really a product of the University of Illinois. He got his degree, I believe, in engineering here.
Yeah.
And was it bioengineering?
No, mechanical.
Mechanical. That's why he can build this thing so well. Anyhow, I don't know if he'll have enough but I love his Cadman drawings. I have learned so much in talking to him. And I really marvel at people who can make an instrument. And that is what Seth is doing. And this is a real achievement. And I was going to ask Ashwin, but maybe you could make a brief comment. It's nice in some ways to have the epoxies, because you really see where it's dark. That separates one cell from the next. But there was also some problems, I know, and you guys were considering different ways of presenting the cells, everything from like the cryo technique of like what Julia Muhammad uses for tomography there. But is there one that is developing along that you're more and more positive about?
Like the sample prep?
Yeah.
Ashwin.
Okay, see, I knew Ashwin should.
So right now the images were shown, and even this image was with a specific type of resin which we do at room temperature, the Kipling and all the stuff. But there's other resins which are done at -40, -20-ish, which
Mhm.
give much better infiltration in these regards. But we
Does it
just
also shift their spectra a bit?
not the cell spectra, but the only problem is those have specific. So in this case, it's really convenient because this epoxy has a band at 1732 where the cells don't. But that
Okay.
different resin has like a slightly different spectra, which is not at 1732.
Mhm.
So in that regards, segmenting out cells could be slightly tricky, but in terms of performance, it's much better.
Mhm.
We just haven't done it yet because it requires basically an instrument which ITG owns, but it's currently broken. But
Story of our
that's
lives,
why,
right?
yeah. So, but yeah, that's why, like,
That's fine.
Yeah.
I just wanted to get an update quickly because I saw at our last poster session, you had some, preliminary results for that. And I really just also want to emphasize that in these experiments, as in anything on biological systems, preparing the samples is so important. You cannot underestimate or overestimate how difficult and what a major component it is to the work. So that is important as finally being able to build a machine to get a signal out of it and have the sensitivity that one wanted, right? Okay, without any further ado, please, Seth, give us the lowdown on the newest version of this wonderful instrument.
Thank you. Just to start, like Ashwin, obviously in charge of kind of the protocol for sample preparation. If anybody here has any interesting studies they'd like to perform, similar to what his talk was, you can contact us. But shown here is basically one of the first results published PNAS 2022 with the original system showing contrasts based on the IR absorption of the different molecules in the sample, showing multiple different colors. Today, instead, Ashwin focused on the actual preparation of these samples. I'm going to focus more on the development of the new system. So my talk kind of split up into 4 sections. First, I'm going to give a little background on IR spectroscopy and near field spectroscopy. So chemical imaging techniques such as infrared spectroscopy provide label-free chemical contrast based on the molecular composition of the sample. So shown here is the typical infrared absorbance spectra of a biological tissue, showing 4 characteristic molecules typically seen and their absorption bands in what's called the fingerprint region. So as you can see here, each molecule absorbs light at discrete infrared wavelengths, resulting in a unique chemical fingerprint. So obviously samples can be then imaged at multiple wavelengths, resulting in a hyperspectral data set with a 3rd dimension equal to the wavelength of the IR light. With the molecular fingerprint of the sample combined with machine learning, this is a very powerful tool for classifying basically the local material content. So conventionally, the infrared chemical imaging data is recorded in a far field microscope configuration using optical system as shown in this schematic. However, like the well-known limitation of this approach is the diffraction limited resolution, which for infrared is particularly worse than what most people maybe invisible would expect, because it's proportional to the wavelength of light, which is much larger on the order of 10 microns. So for infrared, this limits the resolution to a few microns, such as that image there. So for applications that require much higher lateral resolution, we can instead detect the IR signal, not in the far field, but in the near field, configuration using what's known as an atomic force microscope. Conventionally, AFMs use small cantilever probes shown here with a sharp tip, approximately 20 nanometers or so in radius, to map structural details in the height of the sample. And these can be modified in several ways to basically perform chemical imaging. The example shown here is, for instance, just a map of the amide 1 absorption band, which is essentially protein within a single cell, revealing subcellular protein content. So there are many different ways to configure a near-field microscope to detect some IR contrast. Depicted here are 4 of very many different iterations and versions of these systems, each of which are better suited for very specific applications. So for instance, AFMIR is highly sensitive to photothermal expansion of the sample, which is ideal for polymer and biological applications. Because this signals linear dye absorption of the material near the tip, isolating this photothermal expansion potentially offers a quantitative nanoscale chemical imaging technique. But this also has numerous challenges. So hereafter, I'm going to just focus my talk mainly on AFMIR and our modifications for improving single cell applications. So as discussed, AFMIR signal is proportional to IR absorption of the material near the tip because it operates in contact mode, which is probably the only version of these techniques that do that, which operates in this linear regime of the force curve. So any force produces a linear response in the deflection of the cantilever. So early studies of AFMR typically report measurements of very thick samples due to lack of sensitivity early on, which typically result in spectra that are really highly correlated to FTR far field measurements shown in this spectra here. But when you go thicker, the resolution tends to get a little worse because these fields extend deeper into the sample and your resolution gets blurred. So to actually achieve much higher lateral resolution, you typically need some method to isolate the surface measurement signal or basically section samples thin. But when you do that, you need much higher sensitivity because you're reducing the sampling volume. Several reports demonstrate a number of ways to improve the sensitivity of this approach towards monolayer detection with enhancement factors such as electric field enhancements near the tip, cantilever mechanical resonance. So this is just a mechanical amplification of the measurements, or essentially insulating polymer layers with high thermal expansion coefficients. So ideally, we would like to harness basically all 3 of these effects simultaneously to get the highest sensitivity in the measurement. However, in addition to increasing sensitivity, it usually comes with some sort of like artifact or issue, which can be shown here, for instance. So although cantilever resonance enhancement may improve sensitivity by amplifying the cantilever's deflection to the photothermal expansion of the sample, it also is very sensitive to the specific mechanical coupling that exists between the cantilever's tip and the sample itself. So this mechanical coupling can introduce, even randomly, non-chemical contrast and high levels of noise, limiting applications of thin samples and our ability to record really high resolution data. So although this timeline ends at approximately 2017, this is still currently the state-of-the-art approach that the AFMR people use. So to kind of understand the importance of this effect, next I'll just kind of review the AFMR image formation to kind of better understand what the challenges are. So AFMR uses a pulsed infrared laser to induce a small high frequency periodic deflection in the cantilever driven by the resulting thermal expansion of the sample shown here. The amplitude of this periodic deflection shown above is proportional to the local IR absorption of the sample beneath the tip. The signal is then filtered using a lock-in amplifier to recover the first harmonic of this periodic function and isolate it from the DC deflection in order to isolate the signals used for actually height tracking and recording IR data independently within the same signal. So then the sample obviously is then scanned using the AFM scanning configuration to map this IR signal across the sample. So although this image is proportional to the local IR absorption under the tip, it's also proportional to the local cantilever's response shown to the left here. This results in IR images that are actually often masked by structural and mechanical features of the sample, and pretty much due to the local mechanical coupling of the cantilever to the sample. So this has a number of different ways that this can cause artifacts. So although the lock-in signal has a relatively simple form here, the cantilever's response function, which is shown here in purple, is susceptible to these small mechanical coupling changes between the sample and the tip. So this property can vary due to thermal fluctuations in the sample or inherently the mechanical properties of the sample at any given pixel. And this is a very vital aspect of this approach to understand how we can improve performance and sensitivity. So one easy example to show, for instance, is just a plot showing the cantilever's transfer function, which is on the left for a polymer structure that was prepared on a silicon wafer. The two sample locations on and off the polymer show very different frequency response in the cantilever's resonance, resulting from the variations in the mechanical coupling that's seen between the tip and the sample at each location. Collecting an AFMIR image with such a large response and variation can cause a lot of challenges. So here, depending on our choice of which pulsing frequency we want to operate at, we can introduce false IR contrast due to the variability and stiffness seen by the tip, thus masking like the desired underlying chemical contrast. This blue plot here shows just a line profile of the IR signal of PMMA taken at this line on the image. So the expectation here is to see that the signal basically tracks the height of the polymer itself and go to 0 when it is on the substrate here. However, we instead see a rise in signal on the substrate, which is producing basically a signal which would be quantified as a false IR absorption. This results, this can drastically change depending on which frequency you decide to choose. And we will demonstrate later. And in general, it is not something that can be predicted for any general sample, which is not the case for this engineered polymer sample. So another aspect of this cantilever response is that even a stationary cantilever at a single pixel can exhibit similar variations in time due to the thermal perturbations in the system. So the cantilever's response measurements shown here were collected only seconds apart under identical conditions, but reveal observable variations in resonance. So with analytical modeling, we determined that this was caused by small changes in the sample stiffness, which can arise from thermal fluctuations or even small height tracking errors between the cantilever and the sample. So regardless of the reason, even small stiffness changes can have a drastic impact on signal, especially near these resonant modes where you can see the resonant mode shifts just a small amount, but cause a large change in the signal measured in AFMIR. So a relatively easy test is to quantify both the signal and the noise in a measurement at a single point near one of these resonant modes. Here we find that rapid fluctuations in this stiffness, they can introduce noise that exceed the sensitivity benefits expected at resonance. So this kind of dotted line shows the resonant curve. That's sort of what you'd expect for sensitivity. That should scale with the signal to noise ratio. However, the true signal to noise ratio actually dips near resonance with conventional AFMIR. And this is because noise actually scales much worse than the actual sensitivity benefits. So this is kind of one of the primary issues, that there's a trade-off between the sensitivity and this artifact, which creates a lot of extra noise and limits our ability to perform nanoscale chemical imaging of very thin sections and essentially our epoxy embedded cell sections. So here we can kind of see the combined effects of all of these aspects that I've discussed in response in these AFM measurements of epoxy embedded cells. So these three images were all collected at the same IR absorbing band, typical of what you would get for basically protein, the amide 1 band, but collected at different pulsing frequencies, only separated by about 20 kilohertz. And this is conventional AFMIR deflection measurements here. So the expectation here is that each image should provide a reliable measure of the protein content within the cell sections. However, the cantilever response variations are dominant due to the heterogeneity in this sample, not typical of the really smooth polymer targets that we had, but a lot of little structural effects that happen here. So instead, what we could try to do to maybe improve this process is to basically track one of the resonant modes using resonance enhanced AFMR, which is current state of the art in the field, to try to minimize these effects. So here we show results from the current state of the art AFMIR method using resonant tracking. So the images on the left were collected using AFMIR while tracking and pulsing at the resonant frequency at each pixel. Although the intention is to minimize the effects of the cantilever's response, we see little improvement in the chemical contrast when you compare it to the fixed frequency measurements on the previous slide. So besides high noise near resonance, the animation to the right over here shows that the peak amplitude of any given resonant mode is not necessarily invariant to these changes as well. So in this animation, the red line shown tracks the magnitude of one resonant mode as a function of sample stiffness using our analytical modeling. And you can see, depending on the sample stiffness, the magnitude of this peak can still rise and fall, and that would cause these mechanical contributions here. The image on the left is intended to be the epoxy signal, and then the right is protein, but we don't get much distinction between these. So although this plot here is kind of an extreme case, this just sort of shows that the resonant tracking approach is not an effective way to get artifact-free measurements. Our NDIR approach, on the other hand, shows strong chemical differences between the epoxy channel and the protein IR bands. So to first approximation, NDR is designed to produce signal that's invariant to any cantilever response effects. So next, I'll just go through kind of the details of the NDR approach and its benefits for our single cell imaging. So it's important to know that to distinguish between the cantilever effects and the expansion shown here, a constant reference vibration generated by a piezo actuator underneath the sample is needed to basically, you can essentially see it separates the two in some way. It gives us another dimension of data to be able to distinguish between these two. We did this a while ago, and our first approach was basically to collect two separate images with the IR and piezo on independently, and then process their ratios However, this approach fails to improve the noise effects that I was kind of talking about in the previous slides when you operate near resonance. So that's not a great approach to solve this problem. So to understand how NDR improves measurements, let's first consider the signals in conventional AFMR deflection measurements shown in these plots here. So as you can see, the photo-induced heating produces a harmonic expansion signal, which is shown here to the left as this sine wave. In this case, the piezo signal is drawn as 0, it's not on. So following the model, the sum of these two signals are then amplified by the cantilever's response, which we've shown here as a step response. So this can be induced by basically any mechanical change in the system. So in this example, a step change in the cantilever's response that originates from unknown mechanical changes in the system produce a comparable step change in the output deflection, which this results in basically a measurement that's highly sensitive to any response variations, which can occur as a result of mechanical coupling in the sample. So this technique is sensitive to like almost linear proportional to any mechanical effects seen. NDR is fundamentally a new design that's insensitive to these response variations, relying on the destructive interference between the IR and the piezo vibrations. Because the photo-induced and piezo expansions are harmonic, they add coherently, resulting in an acoustic interference behavior seen right beneath the tip. So in this example, when these two vibrations are controlled to have equal amplitude but opposite phase, they destructively interfere, resulting in 0 cantilever deflection as shown to the right. And this is independent of whatever the cantilever's response step function is. So thus any perturbations in the cantilever's response, they have no effect on the cantilever deflection or the system as a whole, since it is scaled by 0 expansion due to the destructive interference of these two waveforms. And this results in basically a measurement that's insensitive to any response variations, including the high levels of noise that we see when we operate near resonance. So in this configuration, we can then recover the IR signal. Instead of measuring the deflection signal itself, which is held to 0, we instead measure the piezo's voltages, which are then proportional to the expansion. So here we kind of just show an example comparison between the AFMIR technique and null deflection. This is just early data taken long ago of a 2-phase polymer. And it's just the absorption band of these kind of beads here. This image is just nice because it kind of shows all 3 of the response effects that can happen while imaging in multiple different areas of this image. So here we show the cantilever's response having numerous effects present in this image. We get mechanical response variations near interfaces typical between 2 different polymers of different stiffnesses. We also get sudden changes in the cantilever's response shown as streaks across the image due to just some change in the cantilever coupling and also slightly high levels of noise in certain areas. So under identical conditions, null deflection isolates the underlying chemical contrast with minimal influence from all of these effects. So the distinction between these 2 techniques becomes even more extreme for the samples that we all care about in section cells because of their heterogeneity. So here we see the influence of NDR for hyperspectral imaging of epoxy embedded cell sections under identical conditions collected for null deflection and conventional AFMIR deflection. So in the conventional AFMIR measurements shown at the bottom, they suffer from these response variations that manifest as a lot of noise and other artifacts at edges and such, masking the underlying chemical contrast that we're interested in. So when combined into a composite image, for instance, on the right, as expected, we see very few chemical features other than maybe a small kind of distinction between the cell material and the epoxy channel in between. However, the top row shows that with NDR, noise and these response artifacts are reduced significantly, and that enables us to basically collect essentially any IR band and overlay them in a chemical image.
So, can you go back? So so how do you get your ground truth here? So your chemical ground truth about where you expect different species to be, because inarguably they look nicer in yours versus the other, but how do you know what's true versus, well, maybe not?
Yeah, that's a good point. We can map it out and get the entire spectra at any given point and isolate it and compare it to FTR is probably a good way. For this study, we basically just demonstrated the performance benefits and kind of made that claim that this is chemical and this isn't. But that's a good
So
point.
when you say chemical, can you help me interpret what I'm seeing in either the separate or composite?
Yeah.
What does blue versus red tell me here?
That's a good point. So the black is essentially epoxy. The blue would be protein, blue, light blue, even green. Blue is basically the amide 1 band of protein. Green channel was set to actually amide 2, which is just another protein band, because at the time we didn't really have many other peaks to look at. The orange and reddish was this other band, 990. And in the paper, we're not 100% certain what that was. It just produced another very unique, distinct contrast. And then when we overlaid them, we could see like a lot of different colors. So at the time, even today, we're still not 100% certain where that peak is. And that's where machine learning in the future will hopefully come in.
Am I right that if you as a proxy for, let's say, ground truth, right? If you just took a panel of standard fluorescence labels for, membrane, cell wall, protein, sugar, stuff like that, that they could serve as together as a ground truth, right?
Yeah. Yeah, we just haven't gotten to that stage yet.
Can I add something? So it's just difficult to do that in the sections what we work in here, because at that level, autofluorescence majorly dominates a lot of it. We've tried like staining, I mean, nucleus and stuff you can obviously do, but if you try to look at like other finer details, then it's just in the sections, it's very difficult to point out something. You can obviously do it in full whole cells, but it's just difficult at like this moment.
So on the same point on ground truth, have you tried any like simpler samples, maybe MMA or other simpler pattern structures?
Yeah.
Just verify how this technique performs.
Yeah, so we've compared to PMMA spectra compared directly to FTR. We have another paper. I don't have it here, but FTR and PMMA show really good agreement to our NDR measurements.
Another technique existing AFMAR. So how much better is the new null deflection method of these control samples?
In terms of like comparison to FTR?
Yeah, comparison to.
We have data. It really depends, but I have another data set that shows like the AFMR technique will have diminished like peaks. They'll get saturated in other ways because null deflection maintains zero deflection. But when operating in resonance, you can see the peaks actually get saturated. So it does show good agreement in certain cases. And I have that data in another paper.
Okay.
Okay. So as for noise, essentially just a small claim. We can get basically up to 24 times improvement in noise and SNR when operating on resonance, where the sensitivity is still, even though it's null deflection, it offers the same sensitivity benefits. So we basically surpass that trade-off between high sensitivity with resonance and high noise. So now I'll just kind of go through the current system design, the prototype we're building to kind of improve the instrumentation. So the current system is just a modified commercial AFMIR, and inside has very slow height tracking, which I've measured, and the IR optics are quite poor, difficult to actually get a really tight focus near the tip, throwing away a lot of our signal. The main issue is the closed loop controls are operated in a software timed input-output operation, and that can only go as fast as a while loop can go in software, which is approximately about 2.5 milliseconds. So this is the speed at which we can operate this. And then obviously there's other benefits, such that like it's very limited in its capabilities. We can't track resonance. The current configuration is figured only for imaging, so we can't go to single spots and take point spectra, which would be really nice. And we can't measure things in parallel, which goes back kind of to the first question of this talk, which was how well do the images get registered? So this is kind of showing what the bands look like after imaging, separated each band in 3 hours approximately to collect a single image. They drift quite significantly, approximately hundreds of nanometers. So our image registration can actually improve that and bring them together to form a nice composite, but pushing the resolution down further, this will eventually be a limitation of the system. Just briefly, I'll talk about this because I think we're getting closer to the time. This is just sort of how the two original and the new system are kind of designed. Because of the complexity of the controls, we essentially have a stability criteria shown here for the closed loop controls. As long as the system's response function falls within this criteria, it is stable and even optimal. But this gain function is a function of the cantilever's response to any kind of expansion. So in real time, we kind of have to measure what that response is at any given pixel so that we can then run the null deflection optimally. The current proof of concept system that we have measures this piezo signal at interlaced lines in a single image. So you can see this does not allow for point spectrum measurements. And then on the subsequent line, it measures the null deflection using the tuning curve obtained in the previous line. So this is kind of how it works. This limits how far you can step. So you really have to have really fine lines and how big of an image you can take with coarse measurements. The new system instead is going to integrate different technology hardware to be able to basically sequence whatever pixel we basically want to command at 50 microsecond pixel rates. So for null deflection, we can then tune with the piezo measurements and then measure null deflection on the subsequent pixel and just have this signal streaming constantly. And then you can just form images and read it out like any other channel. So the current design overview is shown here with the 6 limitations of current AFMIR as of 2023. The system is just a basic microscope body with 2 primary custom components, one being a custom designed AFM with integrated IR optics shown here for improving the optical alignment and focus at the tip. The other is the controller, which incorporates an reconfigurable DAC, which has an FPGA that allows me to basically control the null deflection. So obviously with this type of technology, that FPGA controls, we can operate these measurements at 50 microsecond pixel rates. That's the current rate at which we're running them, which is a 50x improvement over our 2.5 millisecond rate before. We can also stream this null deflection signal, which has the advantage, obviously, it eliminates all the response artifacts that we've talked about, but it also allows us to enable point spectrum measurements, which in the future, I really hope to be able to just in a millisecond snapshot of spectra at any given point you want to measure. Just FPGAs are just great. They basically allow you to sequence this stuff in any way you want, which will also enable us to track resonance. So some samples actually have resonance that shifts at any given pixel. So you actually kind of want to track that to maintain the highest sensitivity at every measurement. Otherwise, certain areas will have really like higher levels of noise. Simultaneously measure mechanical and chemical measurements. So with the piezo measurements and the cantilever's response, we've said that it picks up all these intricate mechanical connections to the sample, but that can be used as a new property map of the sample and potentially help in other ways. And obviously the snapshot hyperspectral data set will allow us not to have to register images anymore. The second major feature is obviously the AFM. It's designed to have about 10x higher bandwidth than the original AFM system that we're using. And higher illumination that can be accessed directly above an ATEC probe, which is an extended probe that extends out. And this will give us a much tighter focus and potentially higher intensity at the tip. And it's all embedded in here, so it's fixed and it shouldn't change per wavelength because it's going to be coming through a hollow core fiber. So no need to align each wavelength independently. And it's very compact compared to the original system, which takes up the size of this table. This little AFM objective can be integrated into basically any microscope system. So just to sort of conclude the talk going through, I'll show some new data images from the new system. But before that, I kind of like want to show this. That was interesting, kind of the theoretical speed limit of the system. So currently the PID is limited by that 2.5 milliseconds at which we can run this simple PID control loop. But what's the ultimate speed limit that we can actually achieve is actually limited by the physics of the cantilever's response itself. So here we show the physical limit imposed by the cantilever. The plot on the right shows an impulsively started expansion signal where the time axis is scaled by the time constant of the lock-in. So here we're just initially starting some expansion signal. So using our analytical modeling of the cantilever transfer function and the lock-in detection, we can model this lock-in's response. So this plot here shows a relatively large lock-in time constant of about 300 microseconds. And you can see no matter what the demodulation frequency is, so the near a resonant mode or just at other frequencies, the signal always kind of responds whatever the lock-in speed is. But if we reduce the lock-in time constant to 10 microseconds, like we're claiming, now you can see very different response behavior. And this was data basically collected or analyzed 4 years ago. This essentially says that our limiting speed limit of the cantilever is approximately 50 to 100, 150 microseconds, somewhere around there, especially near resonant modes, which need a bit longer time to stabilize to the final signal. So with the new system, here's some early data. This experiment is basically just a frequency sweep. Individual pixels are the expansion signal is swept at different frequencies. And in between each frequency, we shut all the signals off. So here we expect to see the cantilever's response and then a function of the dark field, so zero signal essentially. So the blue plot shows the resonant peak, but when we have these pixels at 50 microseconds, you can see that the dark field actually picks up some of the signal. So this is signal that's basically bleeding from one of the measurements to the adjacent pixel. So this is taken with the QCL, the IR laser on a film. You can see if we just delay the pixels by 150 microseconds, this goes away completely and we get independent measurements at each pixel. So right now, this is maybe an interesting direction to understand like how to improve this speed, but that's kind of the rate at which we're going. And this is sort of just the last slide. Data collected with the new system. Currently, we are losing a reasonable amount of power coupling to the fiber, which I do manually. Some of our electronics aren't up to date, and I can get a lot more gain out of them. And our laser diode, we need to upgrade it to a superluminescent diode. to reduce the coherence noise that we're seeing with the deflection laser. All of these effects should boost our performance significantly. But before that, I just wanted to like put a somewhat thick polymer film, so I couldn't get thin section cell samples. But this is basically showing height data, null deflection at a 1732 peak, which is indicative of this polybutadiene spectra. And then this is the polystyrene spectra for this phase-separated polymer film that we published in Analytical Chem in 2018. So this is the exact same sample, and null deflection is producing the signal with the new system. Within the week, I'll probably be able to get the sensitivity and throughput up higher and potentially get some cell images. But I'd just like to acknowledge, obviously, the QCB for funding this work, lab members of the Kissel group, and my collaborators shown here. And I'll just kind of end on this slide showing the timeline of current state-of-the-art AFM-IR systems and their progress to date. But the newest system is essentially exactly the same as everything I've shown, except it is now 700 times slower than our current system. So just comparing the timeframes for collecting kind of some of these cell images we've taken, like 100 micron field of view, 50 bands. Something that would take our new system 3 hours to collect will take the current state-of-the-art system 98 days. So that's where I'll probably leave it. And if anyone has any more questions. Yeah.
So I can sort of easily extrapolate, how this is used in, like a clinical question, right? 6-cell healthy cell, or phenotyping. But how do you? I'm a little I have a gap in how does one use it for, we're here for quantitative cell biology, meaning counting specific molecules. Like what kind of sensitivities we're talking about, like how many nanomolar and how close to species of molecules, protein 1 for protein 2. Can you tell give me a sense of what biological or biochemical knowledge gap I can address here, or even not now, but soon.
Yeah, I mean that's sort of a question for like when we are able to like use machine learning and stuff. But because the system produces a linear response to IR absorption, you can expect like any signal to be proportional to kind of the local concentration of the molecular species just under that tip. So until we get better methods to basically quantify these things, my goal is basically just to get the most pure FTR quality like signal at this nanoscale.
Do you have a sense of the chemical sensitivity, just the ballpark of a ballpark, with a nanomolar, micromolar? What are we talking about? Molecules in the cell of a given species.
Yeah, I don't have a sense of the molar sensitivity. I just know that the signal does come from the molecular species that exists within like approximately a 20 nanometer volume underneath the tip. So you can probably extrapolate that out.
Square or cube? How should I think of it?
It's approximately a hemisphere, like right under the tip.
So a hemisphere of a radius of just tens of nanometers.
Yeah, and there's plots that show like how these fields. So the signal is generated by the electric field enhancements produced by the gold tip, and they'll scatter out approximately the radius of the tip. So it's on the order of 10 to 40 nanometers.
So that's pretty good. So even in a bacterial cell, you get quite a nice set of voxels or something.
Yeah, and we're actively exploring that as like a third like kind of aspect of this system, and we have some good understanding of kind of like the depth sensitivity of where we're getting signal, and we're trying to basically like lock that down to know exactly we're getting this surface sensitive measurement. If you want to add something.
I'll add something. So I think Seth obviously gave a good detailed explanation of what's going on. What is so critical about this work is if you don't have highly accurate signal to noise ratio data from a well-defined volume, nothing else matters. You can do nothing else at all, right? So I think the one aspect is to really push this instrumentation down, carefully controlling and understanding each step of the way, down to perhaps I challenge Seth to 5 nanometer things, because you can get AFM tips now that are just a few nanometer down. So again, that's a question, how quickly we can get there. So that's one aspect, and this is perhaps the most critical aspect of the whole work. The second aspect that Ashwin didn't quite explain, but is part of the second part of Ashwin's project, is to build a database of all known metabolites and their IR spectra. So he's got some, hopefully we can get him to give a whole talk, but he's got some beautiful data on how to, in a high throughput way, measure all the known metabolites and their IR spectra and their distortions, right? So it's a purely measurement exercise. The third aspect is really connecting the theory-based distortions, mixture-based things we see through AI to be able to, when the sampling volume becomes small enough, go and individually identify the metabolites within that sampling volume.
Why do you even have, I mean, to me, ballparks of 10 nanometers or 10-ish is very good and very impressive. Why specifically you need to go even smaller than nanometer range? I mean, if things compare yourself to, fluorescent microscopy or even super resolution, you're already very, very much there.
Yeah,
we don't know the limit. That's a great question, because we don't know the limit, right? So imagine a 20 nanometer hemisphere volume. It has probably tens of metabolites and different concentrations and so on. Now we want to deconvolve all those. So from our prior experience with infrared spectroscopy, if you have a mixture with about 20 components, you're going to be fine. You can actually resolve all those and everything's going to be okay. But once you start getting a larger number than that, you start becoming more and more ambiguous. And then your signal noise ratio requirement to resolve anything just goes through the roof. So there's a connection between signal noise ratio, sample volume, the diversity that you have in the volume, and that's not physics-based, right? So it's not
right,
possible to predict a priori.
Right, but you're, I guess to me what I'm a little missing, but it is like where the biological or biochemical knowledge gap is. So to me, if you tell me you can tell on a 20 nanometer cube, that information, that looks to me already addressing very dramatically knowledge gaps. You're not, you're getting ahead of much of what we know biochemically. I mean, cell biology / biochemistry wise.
You're right. We might. I think the original question that we wanted to probe was those things that you cannot label and measure inside a cell.
Right.
We should be able to directly record.
Sure.
So obviously the wonderful thought experiment is if we could make the tip as sharp as one molecule, we'll basically be able to measure the spectrum of every molecule in the cell, right? Obviously, but that's not going to happen. So there's some finite volume that is practically measurable and practically deconvolvable in its IR spectrum to look at those things that we cannot label. That's what we're after. But the field is so like there isn't a metabolite database in the mid-IR, right? There isn't a way to measure 20 nanometer volumes in any reasonable speed. There isn't a way to connect those two with AI. So again, those are things that all the three students are working on.
Okay, and we can talk on.
So we love your insight and input into this because getting this kind of question at the initial stage of development will perhaps help us pause and solve something, do some experiments, go to the next step, go systematically, right? But this is great. I think Seth was very modest. This is years of work to put this instrument together, and the fact that he has data is we're all celebrating, right? I mean, this is so really incredible amount of work and insight from Seth to get us to this point. But I know that's not a fair table, Seth, but okay, that's where we are.
But I think it's still impressive. I mean, it is the bottom line, it's the time that now you have, is really down to 3 hours.
Right now I just pick random parameters. Like let's say this data
Don't be
set took.
too honest.
I mean, let's say this data set, whatever I defined here, takes us 3 hours. It would take them 98 days. Like that's like I'm wanting to make kind of a figure like this for an actual data set. So I just need to get the system up and like working at highest power. And
I think we should say mapping a single cell. What's the time that it will take? And I think,
Yeah, sure.
you know,
And it's like really important to kind of snapshot these like the spectra at each pixel. So like that's it goes back to your question of like you do these bands separated in 3 hours, but can you really image register them down to nanometer length scales everywhere in the image? Like I don't know if the image registration will be that good if we ever push the resolution limits down.
But the fact that it is going to be much, much faster, whereas what you already, I think, just published in 2022 was taking you much, much longer. So,
Yeah, 2022 data took me like a week to collect the whole hyperspectral.
right, and any improvement on that gets you one step closer to dynamics, you know, but what I sort of missed is the thickness of the samples that you can deal with. Is that at, what did you say, 100 nanometers? Or,
Right now we do between 50 and 100. Actually, don't know if have you measured it? Like I feel I measured once it was 70, but
Yeah. 70 we've tried.
that's typically what we target. Like I said, we're doing a third region of like this project where we're trying to like isolate surface measurements
Sure.
so that we can go thicker and not have to worry about like the resolutions. But these fields extend deeper, so if you just isolate it thinner, you get.
I'm just wondering, you know, like people who do tomography, they also have to do fib milling. And they can get those, you know, they vary, but it's typically around, I think, 200 nanometers. You could mill it even more closely. You could almost think of milling through the cells and then you look at each of those layers.
Yeah,
That would be, you sort of pointed that way in one of your earlier slides, but
like milling it down, image. and
yes,
mill it.
yes.
Yeah, that's like one way. Ashwin's almost made progress on just like doing serial sections.
Okay,
And there are systems that like do that, like you could just do serial sections and then image every section. So like the new system I'll have access to the controls to be able to like identify where regions are and then do whole hyperspectral of every section. And then we can build up like a 3D like image. But that's a ways out.
that'd be genius.
Maybe a couple months.
could you talk 10 seconds about tomography prospects
Okay.
and how the SNR is kind of the critical stumbling block?
Well, obviously, with that serial section method, SNR is not a critical one, like, but you have to be able to like find them and then overlap them. The other one is fib etching,
Yeah.
milling. Then you need a combo fib and AFM, which you really want. I don't know if I want to build that yet. The third one is like computational tomography, which essentially like these fields, this is what he's doing right now, is that the fields extend like into the sample. There's like a 20 nanometer region that's really hot that produces majority of the signal. So they go a little deeper. So at different pulsing frequencies of the laser, that heats the sample and then that thermally diffuses. The length scale is proportional to like the pulsing frequency of the laser. So if you do other pulsing frequencies, you can actually recover signal from different depths and laterally essentially. And then potentially take those measurements, multiple different pulsing frequencies, and invert the frequency dimension to basically recover like depth. But that problem is like either ill-conditioned or ill-posed because we created a new one recently that might be better. And in those types of analysis, noise is a huge factor. So any small error in your measurements, when you try to invert that, just blows up essentially. So getting the highest quality like expansion measurements essentially is what we need with the lowest noise. Then potentially we just take multiple pulsing frequencies and recover like 3D depth down to like a couple hundreds of nanometers with like 10, 50 nanometer resolution or something.
I mean, there's so this interesting mixture of physics, chemistry, and biology in this problem.
Yeah.
Well, thank you again, Seth and Ashwin. Wonderful talk.